Hello, everyone. Welcome to my video tutorial for bisulfite sequencing data analysis. There are other methods to analyze bisulfite sequencing data. Today, in my demonstration, I'm going to show you the HR workflow. So, bisulfite sequencing method is the current gold standard to study DNA methylation. First, let's have a look at how this method works. You can see here is the DNA sequence. We can extract the DNA samples from cells or tissue. Then in the DNA sequence, some cytosine were methylated, some were not. In order to identify methylated and unmethylated cytosine, so we treat the DNA with bisulfite. If the cytosine is methylated, then the cytosine is protected from conversion. But if the cytosine is not methylated, then bisulfite treatment can first convert the cytosine to uracil, then uracil is further converted to thymine. So after the bisulfite treatment, then we can perform DNA sequencing. You can see here is the sequence after the bisulfite treatment. The methylated cytosine will be remained as C during the sequencing, but the unmethylated cytosine will be sequenced as thymine. After the sequencing, then we can align sequencing result to the reference genome. If a cytosine matches to the genome sequence as cytosine, then this one is methylated. If a thymine matches to the reference genome as cytosine, then this position is the unmethylated cytosine. So after the alignment, we can quantify how many methylated cytosine and how many unmethylated cytosine from the sequencing result. So here is the basic method for bisulfite sequencing. You can see it is very easy to understand how this method works. So after the short introduction, we can start our data analysis in R. So for today's video tutorial, we are going to use the dataset GSE 86297. You can download the data for practice using this GSE link. We can have a look at the dataset first. You can see the title for this dataset is Transcription and Chromatin Determinants of the Rate of De Novo DNA Methylation in Oocytes. So in this study, they performed the bisulfite sequencing. You can see down here they have 6 datasets. They sorted the oocyte according to their size. You can see 40 to 45, then 50 to 55, and 6 to 65. So they are in micrometers for the oocyte size in culture. So each size they performed 2 repeats. You can see for 40 to 45 micrometers they performed the sequencing for library A and library B. For 50 to 55 they also performed the sequencing A and B. It is the same for the size 60 to 65. If you download all the data, we will have 6 sequencing dataset here. I downloaded already, so we can go to R and perform the analysis. So first I prepare the text file as a target to show the sequencing data information. So let's read this target in and have a look at the data information. Let's run, and we can click the target, have a look at the data information. You can see here we have 4 columns. We have the GO information in the first column, Then samples in second column under the group, we divided the samples as 40, 50, and 60 micrometers. Then here is the files name we downloaded from the NCBI website. So we have 6 data set here. First, we can load the first data as an example to look at the sequencing data information in each sample. Let's run. You can see we loaded the first sequencing data as sample 1. If we click the data set, you can see we don't have the column information. So let's set the column names. And we can have a look at the data again. You can see now the sequencing data for each sample contains 6 columns. The first column is the chromosome information. The second and the third column are the cytosine position. Here the start and the end position for each cytosine is the same. Then the fourth column is the information about the percentage of methylated cytosine. The fifth and the last column show the information about the cytosine in this position on the chromosome 6 is methylated or unmethylated. Here is the total count for the methylated and unmethylated cytosine in this position. So I use the first data set as an example to show you the sequencing information in the data set. Now we can load all the data set. So there is a function in the IGR package called read Bismarck to DGE. Because the sequencing data is analyzed by the Bismarck package, then we can use this function to load all the data from 6 files as a DGE list. We name it as Y0 here. So you can see the files will be the 6 files in the targets, and the sample names will be the sample names in the targets. I showed you in the beginning the data information in the target file. So let's load all 6 samples as a DGE list. We can run. You can see R is loading all 6 samples. Now you can see in the data windows we have a DGE list data set. We can use the dim function to look at the data set. We can run. Okay, you can see we have more than 2 million system positions in 6 data set. But here you can see we only have 6 data set. But in the DGE list, it shows 12 samples. So we can get the counts from the DGE list to have a look at the samples names. While we have 12 samples now, we can run. Then we can click the counts. You can see the row names are the positions for the cysteines. Then we have 12 columns here. We only have 6 samples, but we have 12 columns now. You can see when we create the DG list, we created 2 columns for each dataset. Contains the count for methylated and unmethylated in the same position. So that's why we have 12 columns here in the DGE list. So next we can get the samples, and we can click the samples. You can see we have 12 row names, group names. At the moment it's all labeled as 1. We have the library size and the normalization factors, and it is 1 for all the normalization factors because we haven't performed normalization yet. And also we can get the gene information from the DG list. We can run. Then we can click the gene data frame. You can see in the DG list, We have the chromosome information and the locus. So next we can use the table function for the chromosome. If we run, you can see down here, we have the total numbers in each chromosome from chromosome 1 to 19. Then we also have the X and the Y chromosome, and we have the mitochondria DNA information as well. Because we are not going to analyze the mitochondria DNA methylation, we can remove the count for the mitochondria DNA. We can remove it. If we look at it again, you can see now it is 0 for the mitochondria DNA. And you can see here as well, the chromosome is not in the order from 1 to 19. So we can reorder the chromosome for the dataset from number 1 to 19, then X and Y. So let's reorder the chromosome information. Now, if we use the table function again, you can see now we reorder the chromosome from chromosome 1 to 19, then X and Y. Also, we don't have the mitochondria DNA information here now. Okay, until here we loaded the sequencing data, and also we reordered the chromosome names. So next we can annotate the CpG site. Use the gene names near to the CpG site. To do so, we need to search for the gene transcription start site, then determine the distance for each CpG site from the closest transcription start site. So we can use the nearest transcription start site function to annotate the CpGs. We can run. So you can see we created a new dataset named as TSS. Let's click this dataset and have a look at the data information in this new data frame. Let's connect it. You can see the first column is the gene ID, second column is the gene symbol, the third column is the gene wise. Then the first column is the transcription start site for this gene. Then fifth column is the strand information. And importantly, the last column shows the nearest distance from this transcription start site for each cytosine in our dataset. Because the TSS is a separate data frame, now we can add the information from the TSS into the DG list. Before we do that, we can have a look at the gene information I showed you in the beginning. We have the row names for the position of each cytosine, and we have the chromosome information, then the locus. Now we can add the gene ID from the TSS data frame to the DG list. Next is the symbol information, width, strand, and the distance. Now we can have a look at the genes slot again in the DG list. You can see now we added the gene entrance ID, symbol, width, strand, and the distance information in the DG data list. So after the annotation, we can filter in the data and also normalize the data. When we created the DG list, we separated the methylated and the unmethylated cytosine in the same position into two columns. In order to get the total count for each cytosine, we need to add the methylated and unmethylated count for each cytosine together. First, we can get the methylated and unmethylated count for each cytosine from each samples. Let's get it. Then we can get the count for methylated, unmethylated. Then we add them together as coverage. if we look at the coverage data frame, you can see now we only have 6 columns. We have the total count for each cytosine in 6 different samples. So after we got the total count, now we can filter the data. So as a general rule for filtering the data, we only keep the total count for both methylated and unmethylated cytosine bigger than 8. So we can use 8 as the threshold for the count and have a look at how many have more than 8 counts in all 6 samples we can run. Now we can use the table function to have a look at how many have the total count bigger than 8. You can see here we have 373,697 have the total count bigger than 8. Because we are going to analyze the differential methylation site in 3 different sample sites, 40, 50, and 60 micrometers, we can remove the cytosine in the dataset that are always methylated or never methylated, because those cytosine sites is not useful for us during the analysis to identify differentially methylated cytosines. So let's get the information for always methylated and also always unmethylated from the dataset. We can use the table function again to look how many cytosines meet this requirement we can run. You can see we can only keep 550,803 for our analysis. So now we need to combine both requirements for the filtration. So let's use the table function again to look how many cytosines meet our requirement for both conditions we can run. You can see now we only have 254,912 cytosines that we can keep for our analysis. So now we can filter the data, use the requirement we set up here. Let's do the filtration. We filtered the DG list and created a new DG list as Y. After the filtration, we can have a look at the YDG list. You can see now we only have 254,912 cytosines for our analysis, and we still have 12 columns. So after filtering in IGR analysis, we normally perform normalization. According to the IGR protocol, we don't need to do normalization here, but we need to set the library size as the same number for both methylated and unmethylated in each sample set. Before we do that, we can have a look at the library size again in our dataset. So you can see for the first sample, 40 to 45 micrometers. In the dataset A, we have the methylated and unmethylated. You can see the library size. Generally, the methylated library size is smaller than the unmethylated library size. Even we created 2 columns in the DG list, but for the analysis, we are going to analyze the methylated and unmethylated data as one sample. So we need to set the same number for the library size for methylated and unmethylated in each dataset. So we are going to use the average library size for methylated and unmethylated library size for each sample. So what we can do, we can add the library size, methylated and unmethylated together. The total library size we can run. You can see now here is the total library size for methylated and unmethylated cytosines. in each dataset. Next, we can divide it by 2. You can see now it's the average library size for each dataset. Then we can set the average library size for each samples. We can run. Now we can have a look at the library size in each samples again. You can see now for the first sample A, you can see we have the same library size. For sample B, you can see methylated and unmethylated cytosine have the same library size. So that's all we need to do for the normalization step. We don't need to do more normalizations. So now we can have a look at the data. We can make a MDS plot for each samples because we have the count for the methylated and the unmethylated cytosines in each data set. So we are going to calculate the M values for each data set for the plot. So the M values is log 2 methylated, take away log 2 unmethylated. Because we want to avoid the zero count, then we add the value 2 for the calculation. So first we get the count for methylated, then unmethylated. Now we can calculate the M value. We can have a look at the M value for each data set. You can see the row names are the cytosine position on each chromosome. The column names are the sample names. We have 6 samples here. So now we can get the column names to label the samples on the plot. Let's get the column names. So we have the column names as the same name in the target samples. So now we can make the MDS plot. We can run. Let's zoom in. You can see we have 3 different sizes for the samples. 40 to 45 for A and B. They are on the left hand side. 50 to 55 for A and B. They are in the middle. And 60 to 65. They are on the right hand side. You can see from the distance from all the samples, sample size 60 to 65 are highly reproducible from A and B. And also for the sample size 40 to 45, they are very close. But for the sample A and B, for the 50 to 55 micrometers samples, the distance is quite far. So for our demonstration later, we will compare the 60 to 65 with 40 to 45. Because the data from 60 to 65 and 40 to 45 show less technical variations. So now we can design metrics to perform the rest analysis. So first we can design the matrix to contain all the group information. We use the 0 plus here to include all 3 groups. Let's design the matrix. If we click and have a look at the design matrix, you can see here we have 3 groups. Group 40, group 50, and group 60. So here we only have the group information. So next we need to expand the matrix design to identify the methylation effect in the dataset. In Agile, there's a specific function for DNA methylation analysis to identify the methylation effect. So we can use this function to expand the matrix design. If we have a look at the design matrix now, you can see we have 6 columns to include the sample information from sample 1 to sample 6. And also we have 3 columns to include the group information. So now we can use the matrix to estimate the dispersion. Because we have a bigger data set here, including methylated and unmethylated cytosines on each chromosome from chromosome 1 to 19 plus X and Y chromosome. So in order to save the time, we are going to use chromosome 1 for the demonstration. First, we can subset the data for all the cytosines, methylated and unmethylated on chromosome 1. Let's subset the data. Now we can use the design to estimate the dispersion. For DNA methylation analysis, previous study showed there is no relationship for the trended dispersion, so we are not going to estimate the trended dispersion. We can set the trended dispersion as none. Then we can estimate the dispersion. If we look at the data for the estimated prior degrees of freedom, you can see they show infinity for all the CpG sites, which means all the CpG-wide dispersions are exactly the same as the common dispersion. So after the estimation of dispersion, we can perform differential methylation analysis at the CpG loci between different groups. First, we can fit the gene-wide linear models. Let's fit the model. As I mentioned, we are going to compare the group 60 micrometers against the group 40. We can make a contrast to compare the group 60 against the group 40. So now we can perform the likelihood rate test. Use the contrast we set for group 60 against group 40. Let's perform the likelihood rate test. After test, we can use the top tag to look at the top differential methylation CpG site. Let's run. So you can see here we show the top 10 cytosine sites that show differential methylation. And we can summarize how many cytosines are methylated and how many are demethylated between group 60 micrometers and group 40 micrometers. Let's run. You can see we have 24 cytosines were demethylated and 1,738 cytosines are methylated in the group 60 micrometers against the group 40 micrometers. So finally, we can make a plot to show the cytosine methylation and the demethylation. So we can run the code to make a plot. You can see we generated the plot, compare the DNA methylation between group 60 micrometers and group 40 micrometers. So the red dot show the cytosines that increased the methylation in the 60 micrometers group, and the blue dot show the cytosines that have decreased the methylation site in the 60 micrometers group. Okay, so that's today's demonstration. I showed you how to use IGR package to analyze the bisulfite sequencing dataset. In my next video tutorial, I'm going to use the IGR package again and show you how to examine the methylation levels within the gene promoter region. So please subscribe my channel and share my videos with your friends. I hope to see you in my next video tutorial.